Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is for 14th October 2020. As we have been into the revision mode, in our previous session we were talking about pteridophytes and we classified pteridophytes also. In this session we are going to talk about another group called gymnosperm. So the gymnosperms are plants in which ovules are not enclosed by any ovarian wall and remain exposed. That's why we say gymnos means naked, sperms means seeds. Plants that are having naked seeds, they are gymnosperms. The seeds that develop post fertilization obviously are not covered. That's why naked seeds. So gymnosperms, you must remember, are the plants that have naked seeds, and they are perennial and woody. A lot of wood will be there, forming either bushes or trees. Some are very large trees like sequoia, okay, and others are very small like jambia. So you got very large plant and very small plant. Stems may be unbranched like in cycas or branched like in pinus. Root is tap root. Leaves may be simple leaf or compound leaf. They are heterosporous. Hetero means different. Sporous, spores. So spores are not similar, dissimilar spores. Heterosporous. Produce haploid. Haploid means one set of chromosomes, n number, microspore and megaspore. So there is haploid microspore and haploid megaspore. Haploid microspore in male and haploid megaspore in female strobile. There is structure called strobile bearing this. So male and female gametophytes do not have independent free living existence. Pollination occurs through air and zygote develops into embryo, and ovules are seeds. So if someone asks you what are seeds, your reply should be, as being a biology boy, developed ovules are seeds. And what's a fruit? Developed ovary is a fruit. And the seeds, developed ovules, are naked. That's why the group is called gymnospermae. Plants possess taproot system, but in some forms coralloid roots also will be present. What are these coralloid roots? Roots having endosymbiotic with blue-green algae, like example cycas or mycorrhiza, that is pinus, may be present. The stems are aerial, erect, woody, branched like in pinus, unbranched like in cycas. Leaves are usually dimorphic. Again, this is a technical word, dimorphic. Leaves are of two types: large green foliage leaves and small brown scaly leaves. So that's why dimorphic. Di means to morphic, morphology, external appearance. Xylem does not possess vessels except in some gnatophytes, that is gymnophytes. Phloem is without companion cells and sieve tubes. Sieve cells are not arranged end to end in rows. Vascular tissues are arranged into vascular bundles. Vascular bundles of stem are open, means having cambium in between xylem and phloem. So there will be, like cambium is like a circle, there will be xylem and down there is phloem, in between there is cambium. So you got a xylem, phloem, in between cambium, then we say open bundle, open vascular bundle. So that secondary growth is quite common because of the presence of cambium. Secondary growth will be there. The gymnosperms are heterosporous, as I said just now, microspores and megaspores. Haploid microspores, haploid megaspores. The two kinds of spores are produced within sporangia that are borne on sporophylls, which are arranged spirally along the axis to form compact strobili. And the strobili, if you remember, recollect from your memory, we call them as cones. Two types of sporophylls, microsporophylls and megasporophylls, are usually aggregated to form distinct cones or strobili, pollen cones and seed cones respectively. So pollen cones, if you remember, male cones, and seed cones are female cones. The microspores develop into a male gametophytic generation which is highly reduced and is confined only to a limited number of cells and completely emerges out from the microspore. The reduced gametophyte is called pollen grain. Ovules are naked and are borne on the megasporophylls. Each ovule has a mass of tissue called nucellus. So this nucellus has got its significance. Nucellus is a nutritive tissue. So it is equivalent to megasporangium, and in that MMC will be developed. What is that MMC? Megaspore mother cell will be developed in that. So basically the pollination, pollen grains are directly carried by wind to the micropyle end of the ovule. Therefore, only anemophily occurs. And then of course there is polyembryony, that is development of several embryos in one seed, of which only one survives. And the number of cotyledons may be 1 or 2 like cycos. Okay, and gymnosperms are divided into 4 groups. Let's see, and they are Psychopsida, Ginkgopsida, and Coniferopsida, and Nitopsida. And with that, we move on to another group of plants, Angiosperms. Of course, for your batch, this is not there. This is deleted. Yet, we'll talk about this in a brief so that maybe in your NEET exam or some other national competitive exams, this will be of use to you. Particularly words like synergids, antipodals, double fertilization, particularly all these come in this angiosperms. So it is better just these at least words. That is, pollen grain and ovules are developed in specialized structure called flower. So first time flower has come, flowering plants. That's why angiosperms are called flowering plants. Seeds are enclosed inside the fruits. Just now we said, what are seeds? Developed ovule is a seed. What about fruit? What's a fruit? Developed ovary is a fruit. Size varies from almost microscopic Ulfia 0.1 centimeter to tall tree such as eucalyptus, more than 100 meters. In our campus also many are there, eucalyptus. Okay. The male sex organs in a flower is the stamen. It contains pollen grains. The female sex organ in a flower is pistil or carpel. Pistil consists of an ovary enclosing one or many ovules. Within ovules are present highly reduced female gametophyte termed as embryo sacs. Each embryo sac has 3-celled egg apparatus, 1 egg cell plus 2 synergids, so 1 plus 2. 3 antipodal cells, 2 polar nuclei. So totally how many? 1 egg cell, 2 synergids, 3 plus 3 antipodal, 3 plus 3, 6 plus 2 polar nuclei fused, so 7-celled. So that's why it is called 7-celled 8 nuclei body. The polar nuclei eventually fuse to produce a diploid secondary nucleus. So these are the things that help in double fertilization. So basically angiosperms are those seeded plants in which seeds are formed inside the fruits and the sporophylls are organized into flowers. Angiosperms are the most recently and highly evolved plants. Xylem contains vessels phloem possess sieve tubes and companion cells. Secondary growth occurs in stem and root of some angiosperms placed in group dicots. That means we are going to divide again angiosperms based on evolutionary trends into monocots and dicots. Sporophylls are highly modified structures. A microsporophyll or stamen consists of a filament and an anther. A megasporophyll or a carpel consists of stigma, style, and ovary containing ovules. Within ovules are present highly reduced female gametophytes known as embryo sacs, which are seven-celled. This you must remember, seven-celled, 8 nucleate structures. So 7 cells are there, 8 nuclei, 8 nucleated embryo sac. This is very important. Remember, this will help in double fertilization, and this is a major question, 5 mark question. So you try to recollect, go back to the video that I have posted in the month of July, draw the diagram and have a clear concept about it. Embryo sac formation is preceded by meiosis. So all the cells are haploid. Each embryo sac has a 3-celled egg apparatus. So we are talking about the 7 cells. Out of the 7 cells, 3 cells are egg apparatus, 1 is egg cell and 2 are synergids. 3 antipodal cells and 2 polar nuclei. The polar nuclei eventually fuse to produce a diploid secondary nucleus. Pollen grains or microspores reach stigmatic surface found at the tip of the carpel or megasporophyll. They germinate on the stigma, and resulting pollen tubes grow through the tissues of stigma and style of each of the ovule. Here I must make you to recollect our experiment number 1 in the practicals. Growth of germination of pollen tube, remember? So that is experiment also will be there related to this, how pollen tube grows. After the pollen tube reaches the embryo sac, its tip dissolves and the male gametes are liberated. Of the 2 male gametes, one fuses with the egg cell of the embryo sac, that's called syngamy. That is male, we can say, I can say pollen grain plus egg cell, resulting in formation of zygote, that's syngamy. While the other fuses with the secondary nucleus, that is 2 nuclei are there, no polar nuclei, 2 plus pollen grain, that is male nucleus 2, I can say. Male nucleus 2 plus 2 nuclei, triple fusion. And the triple fusion results in formation of pen, primary endosperm nucleus. Pen develops into endosperm, which is the nutritive tissue. That's why syngamy plus triple fusion. If you recollect from your memory, I wrote S for syngamy, and triple fusion TF is equal to double fertilization DF. So double fertilization is syngamy plus triple fusion is double. Syngamy results in the formation of zygote. Triple fusion results in the formation of pen, and pen develops into N. I can say E, endosperm. So endosperm is the nutritive tissue. That's why double fertilization. The synergids and antipodal cells disintegrate after fertilization. Product of triple fusion, as I said, is endosperm, which is triploid structure, 3N, because it is triple fusion, so it has to be 3N. Whereas this is zygote is 2N. And endosperm provides nourishment to the developing embryo. So this is a developing one. And the angiosperms, based on the evolutionary structures, are divided into 2 groups further, monocots and dicots. So class monocotyledons, class dicotyledon. Mono, as the name suggests, 1, cotyledon, seed cover. Di means 2, cotyledon, seed cover. So single cotyledon, 2 cotyledons. Parallel venation, reticulate venation. What is parallel venation? You know, just like railway lines, parallel venation. And reticulate is a network-like thing, right? Fibrous root, taproot. Closed vascular bundle, open vascular bundle. Just now I said, what is the difference? Presence of, what is the tissue we said just now? The presence of cambium we said. More number of vascular bundles, many will be there, freely distributed, less number of vascular bundles. Example, monocots, banana, wheat, rice, and dicots, grams, mango, apple, all these dicots. And double fertilization, each pollen, just now I said double fertilization, each pollen can produce 2 male gametes, one gamete fuses with egg to form embryo, this is called syngamy. Other gametes fuse with 2 polar nuclei to form endosperm, triple fusion. Since fertilization takes place twice, it's called double fertilization. And then alternation of generation, next concept. What is that alternation of generation? Different plant groups complete their life cycles in different patterns. Angiosperms complete their life cycle in 2 phases, a diploid sporophyte and a haploid gametophyte. The two follow each other. This phenomenon is called alternation of generation. Alternation, one after the other. So one generation is sporophytic generation, another is gametophytic generation. So life cycle, like sporophyte goes to gametophyte, gametophyte backs to sporophyte, life cycle gets completed. In that diploid sporophyte, so 2n sporophyte, and gametophyte haploid, single, one set of chromosomes. And haplontic, sporophytic generation is represented by only one-celled zygote. Meiosis in zygote results into haploid spores to form gametophytes, which is the dominant vegetative phase. Example, Valvax spirogera, that's in algae. Diplontic life cycle, diploid sporophyte is dominant, independent, photosynthetic plants. The gametophyte is represented by a single to few-celled. All seed-bearing plants fall under this category. All seed-bearing means gymnosperms and angiosperms. Haplodiplantic, both phases are multicellular. Intermediate condition is present. It is present in bryophytes and pteridophytes. So with this, we are able to complete chapter 3 revision. Keep on practicing diagrams in this chapter. The life cycles, alternation of generation, one of the life cycles you have to draw schematic sketch. So practice all the 3 schematic sketches. Since angiosperm is deleted for you, focus on bryophytes and then the pteridophytes, bryophytes and pteridophytes. And then since angiosperm is deleted for your batch, you need not worry about that, but these 3 alternation of generations will be there. So keep on practicing, understand the concept, and if doubts persist, post on our group.